thanks, Edna, very much for the invitation. Normally I'm always extremely flattered and honored to be invited by graduate students in giving a seminar at a university, but it was especially nice to be invited by an undergraduate too. So it really is a compliment to be invited for this seminar. How is the microphone? Okay, it's echoing a little up here. I can see. So today, as Edna mentioned, my background actually originated in conservation biology and the study of large organisms. And so today I hope to take you through some of the work we've been doing in one ecosystem nearby Irvine, about half an hour away from campus, to try to, I guess, impart upon you how my lab is thinking about bringing general ecology ideas to microbiology, but also vice versa from microbiology back to community ecology. And so I got interested in microorganisms in part because we know quite a bit about large organism diversity and how it's distributed across the globe, where at least at the time when I was finishing my PhD, we had absolutely pretty much no idea how microorganisms were distributed on land. We have a little better idea in the oceans, but we have no maps such as these where to look at sort of basic diversity, compositional patterns of bacteria, fungi, viruses, and other microorganisms. So one sort of study that came out just a few years after I got into microorganisms was one that just simply estimated prokaryotic diversity, all of bacteria and archaea in samples. You'll notice here that there's no error bars. So this was literally one sample from all these different environments at the time. And this, you may not be able to see it behind the podium, is 2002 that this study was published. And actually, this pattern is kind of held up where probably our most diverse environments that we see for bacterial diversity are sediment environments. Soils, of course, are very diverse, and agricultural soils or very extreme environments like salt ponds have relatively low diversity. So I found this quite exciting that there might actually be some patterns out there in terms of diversity of microorganisms. Now, 10 years later from that study, but still only a couple of years ago, folks like Noah Fearer and his group are publishing research which shows the distribution of bacterial communities across plant biomes, and that the composition of these communities is very distinct, as an ecologist might imagine they would be. But still, there's quite a bit of low-hanging fruit out there in the microbiology world, just showing that there are biomes of bacterial communities. There's also, just to be fair to the marine folks, there are also maybe some marine microbiome or marine biomes where you have differences, for instance, between pelagic, benthic, open ocean, coastal ocean. We see very distinct communities in different parts of the world, in different habitats. Even in the human microbiome, now patterns are being observed. Just in 2013, this nice study came out looking at richness, in this case, on human skin. So if you're looking for high diversity of bacteria, you should look for it on your hands, and high diversity of fungi will be on your feet, evidently. So, but I thought these are great patterns, right? It's just the biogeography of humans, of natural ecosystems, and it's pretty exciting. We're still just learning the basic patterns. Now, the next question any self-respecting ecologist would ask is whether or not these patterns, these differences in diversity and composition, matter to the functioning of ecosystems, or even human ecosystems, natural and human ecosystems. So we know that microorganisms are incredibly abundant. And again, just think about the bacteria and archaea. There's this nice back-of-the-envelope calculation done just before 2000, asking how much of the global nutrient pools are held up in microorganisms, in bacteria and archaea specifically, compared to plants. And so just these numbers in terms of where the nutrients are, you see that in terms of excuse me, carbon, nitrogen, and phosphorus in living organisms, a lot of it is held up in microbes. Okay, but that's just abundance. What about metabolic diversity? Of course, microorganisms are incredibly metabolically diverse, so all plants would fit in the photoautotrophs and the animals would be in chemoautotrophs, but we also have, and I've only put up several other examples of entirely different metabolisms that bacteria and archaea can perform. So certainly an ecosystem would change if we lost all ammonia oxidizing bacteria and archaea. We would expect functioning to change. But here I'm talking a little more about within one of these metabolic guilds, if the composition of the taxa within one of these guilds vary, does that matter for functioning? So in larger organisms, how would we test whether diversity affects functioning? Well, you might do so like at the LTER experiment at Cedar Creek, where you have nice plots and you manipulate plant diversity or composition and go back after several years and measure some measure of ecosystem process rate. And I do not want to belittle this project, which is a huge, enormous undertaking and requires, 50 undergrads a summer to weed and maintain this plot. But we also don't have the equivalent sort of ability to do this for microorganisms in the field. So we have to think about other ways we might test this question. So there's a good reason we haven't been able to test it so far. All right, so several years ago, my colleague Steve Allison and I wanted to think a little more carefully about this pervasive view that microorganisms the composition of microorganisms doesn't matter to ecosystem functioning. And so when we started thinking about it, we realized there were three separate things assumptions that we were hearing from people, usually microbiologists, but some ecologists too that were studying process rates. And so we were thinking about this in terms of environmental change. So a lot of, in the previous slide when I show you the Cedar Creek experiment, a lot of this research in community ecology about diversity functioning relationships has been driven by the need or the desire to understand what might happen as community composition shifts in the face of global changes that we expect to happen. So at the same time, we saw that there's few studies or papers about whether or not microbial diversity would change and whether that would be important. So there are three reasons that we came up with sort of assumptions. And the first is that that microbes might not be sensitive to environmental change. And so that, in other words, to flip that around, they might be resistant, and so composition would stay the same. And there's several reasons why this might be the case. One of the main ones that we were hearing was, well, microbes are really phenotypically plastic. So, I think for the rest of the talk, when I'm saying microbes, usually I mean bacteria. So I don't want to offend anybody else that's studying other microorganisms, but for shorthand, I'm going to use that. So a lot of bacteria are very phenotypically plastic, so your composition may not change because they can quickly acclimate to the new conditions and your composition would stay the same. But let's say that they're sensitive and not resistant, because actually now, since there is a lot of evidence that this is not the case, that communities do shift in response to the environment. So let's say you do have altered composition. Well, and the second argument is that they might be very highly resilient. And so yes, you do see a shift, but very quickly, microbes have the ability to grow really fast, have short generation times, high population sizes. So really quickly, after this change, they'll come back to the composition that they had before. And this could be because of evolutionary adaptation, allowing for the community compositions to stay the same. Okay, but again, assume that your community is not entirely resistant and composition still remains altered after some time. Then the final assumption is that microbes are very functionally redundant. And so even if your composition shifts, the functioning of that community will remain the same. So only if your community is sensitive, not resilient, and not functionally redundant would microbial composition matter to ecosystem functioning. So this is sort of three different things that we needed to address to ask this question. So to get at all three of these aspects, we wanted to use an experiment that had to do with global change manipulations. But you could think about these ideas in terms of in any environmental context and not necessarily in a global change context. And it could just be a manipulation, even in the lab, of some abiotic parameter. But for our purposes, we wanted to take advantage of this really great global change experiment that is led by Mike Goulden, who's in the Earth Systems Science Department at UC Irvine. And so he set up this experiment in both coastal sage scrub and grassland ecosystem nearby. They're right next to each other. And as you can see, he manipulated precipitation. So he had, here you see the rain covers on some of the shelters at the experiment. And so right before it rains, a big storm, in about half the storms, his lab goes out and covers these shelters and prevents the rainfall. They actually collect that rainfall and add it to other treatments that then get increased rainfall, but I'm not going to talk about those plots today. But then they take down the rain shelters right after the storm. So we don't think there's a major effect of the rain shelters themselves. Of course, this can only be done in a place like Southern California where we have like 6 storms a year, so it's not too hard. But even that, it takes the grad students all day to put up these giant shelters. The other aspect he's manipulating is nitrogen deposition. So change in precipitation is something that's supposed to affect Southern California in the near future. And nitrogen deposition, we already have a lot of atmospheric nitrogen deposition in our ecosystems, and that is likely to increase as well. So a group of microbial ecologists, myself included, at UC Irvine, so Kathleen Treseter, Steve Allison, Adam Martiny, myself, and also Owen Brody at the Lawrence Berkeley Labs, we got together and asked Mike if we could parasitize his experiment, right? Because he has these giant plots, and we're like, we just want to take a little bit of sample here, and we won't disturb your plants. And he was amenable to that, and so it's because of his goodwill and a lot of work that we're able to do this experiment. And so we wanted to focus in on one ecosystem process that we thought we could quantify really well, and that was also very connected to the microbial community. So we chose to look at leaf litter decomposition. And so leaf litter decomposition is important in many terrestrial systems, where it's sort of this process that is right at the intersection of whether the plant material, when it dies, is it going to be respired to the atmosphere, or is it going to be stored in the soil as more recalcitrant compounds. It also is a really nice system to work on because we can do a lot with leaf litter that we couldn't do with soil, for instance, in terms of manipulations, and you'll see that in a couple more slides. Okay, so on the right is just a map of Mike's plots in the grassland ecosystem, and he has 8 different blocks, and we chose to look at 3 different treatments: the control ambient conditions, added nitrogen, and the drought plots. And we didn't look at this in a factorial manner, so I'm going to be talking about the experiments kind of as 2 experiments: the drought experiment and the nitrogen experiment. And we sampled the work I'm going to be talking about. Initially, we sampled all 4 seasons for 2 years. And we initially surveyed the communities with 454 sequencing of ribosomal RNA genes. So for the bacteria, 16S, and the fungi, 28S. So I'm going to skip over some of the methods and go right into what is going on with these communities in these plots. And my student, Kristen Matalik, led up this part of the analysis, where she was just taking litter samples from the plots and characterizing the composition, both of the bacteria and the fungi, on leaf litter. And so what I'm showing here is an NMDS plot, and the points are colored by treatments, ambient, drought, and nitrogen addition. And kind of, if you squint, you can see the nitrogen, or the drought. The yellow drought plots are a little lower than the other plots, but there's not a huge signal. It is significant by treatment, but it's a small signal. And this is for the bacteria, but we see the same pattern for the fungi. So now if I plot the same points again, but I color code them by season, you see a different story. So this is just 1 year of data, and you can see these seasons, the date we sampled is really distinct. And so I bring this up because there's a lot of variability in our communities. They're very dynamic, but they're dynamic over season, and that generally is overshadowing what is going on with the treatments. So if I can plot this in a different way, where I'm plotting here an estimate of the % variation in all the composition of bacteria that we're seeing, and how much is attributed to either time or treatment. And so year and season account for a lot of the variation we see. Treatment is less than 5%, the drought treatment explains in the variation. And there's an interesting interaction between treatment and season. And then about half of our composition we can't explain, which actually is pretty typical for any ecology study. We have a review that actually comes up with it's 50% in microbes and it's 50% we can't explain in plants and animals. So I was quite surprised. That's very similar. Anyway, if we compare then the drought experiment or the drought treatment to the nitrogen treatment, we see a very similar pattern. In that, I want to point out that the treatment is significant and it explains a small amount of variation, but not very much compared to the temporal variability. Okay, and just to compare a little bit the bacteria and the fungi, we see the same pattern in the fungi, where here for the drought experiment, you see again, most of the variation is due to temporal effects, and then you see a small amount of variation due to the drought treatment. And I'm going to focus for the rest of the talk primarily on the drought experiment, and I'll come back to the nitrogen experiment at the end, but it's just too much to fit all in one example. And we see stronger effects of drought than we do of nitrogen, so that's the reason for my focus. Okay, so in addition to looking at community composition, we did a few other assays in sort of the survey of the plot of Mike's plots. One thing is we looked at extracellular enzymes. So these are enzymes that bacteria and fungi are excreting and are responsible for degrading polysaccharides in the leaf litter. And so they have often been used then as a metric of the functioning of the microbial community there. But we actually didn't see any effect of drought on the enzymes, although they did vary again temporally. And if we look in the plots at decomposition rates, this is just an example for 1 year where you see there's an effect. So this is % mass of the leaf litter in a litter bag remaining. There is an effect of drought is slowing decomposition, but actually by the end of the dry season, the drought treatment has caught up and lost about the same amount. So not huge. Not huge functional effects that we observed in the drought, not huge functional effects of drought on the functioning of these communities. Okay, but drought can have numerous pathways for affecting decomposition, and one possibility is that we're seeing multiple effects through different pathways that are kind of canceling each other out. Okay, so drought can affect the plant community, which it does, and I'm not going to show you that data. That's from Mike's lab. And it could have another direct effect on what I'm going to term microbial physiology. So you need water for these enzymes to function and for them to degrade. And so if you don't have water, you would expect slower decomposition rates. Now the microbes sort of end up in, the community of microbes ends up in the middle with these interesting indirect interactions that might be going on. And so we do see that the microbial community is changing, but we haven't figured out whether it's changing because it's responding directly to the drought, or is it responding to the difference in the plant community that's now making up the leaf litter. And so that's responding to that. And finally, we haven't considered at all whether or not that microbial community, the shift in microbial community, is ultimately having an effect on the physiology there, and then and therefore decay rate of the litter. So to get at these different indirect effects, we really needed to do a manipulation where we can manipulate the plant community in terms of the leaf litter available, the drought environment that is there, and also the microbial community. So to do this, we used what we are calling our microbial cages. And these are just leaf litter bags that are made of nylon mesh that prevent the migration of bacterial cells into and out of the bag, also fungal cells. And so what we can do then is within each of our cages, we can manipulate all three of those factors I mentioned. So for instance, down here in the precipitation experiment, we have the letter, which is the microbial origin. We have different leaf litter, which could either come from an ambient or a drought plot, and then we can manipulate the microbial origin, whether or not we took the community from an ambient or a drought plot. And then finally, once we inoculate the sterile leaf litter with the microbial community, we can place it either in ambient or drought conditions. And that way we can have all of the different factors there and ask which of these factors is more or less important for decomposition rate. So these are just a couple of pictures of the reciprocal transplant experiment and how it looks next to some of the plots when they're open and closed. And I'm going to give you three questions, go through three different questions with this experiment because it blows up to be a pretty big experiment, and so I want to keep myself focused here. So the first question I'm going to ask is whether or not the shifts in microbial community composition associated with drought, actually I would like that to say drought, so do shifts in microbial composition associated, in this case with drought, do they affect decomposition? And so that to me would be answering that question of are these communities functionally redundant, the communities in the ambient and the drought plots. We see there's a compositional difference. It's not big, remember, because the temporal variability is really high. But there is a difference in composition. Does that affect functioning? Okay, so this question then is one we're answering in the first year of the experiment, because now we've transferred the communities, we put them on the different types of leaf litter and put them out in the plots. What happens to decomposition? So here I'm plotting mass loss. So it starts at 100% of leaf litter, so 0 mass loss, and increases over time. And you see that, not surprisingly, the plot environment affects mass loss. In the drought, again, it's slower than in the control plots. Okay, so that was expected. There was no effect of leaf litter origin, so I'm not going to plot that. So it didn't matter whether you put drought litter or ambient litter in the litter bag. But it did matter if you put different microbes. In fact, it mattered pretty much in the same degree of magnitude as it mattered which plot you were sitting in. So if you had drought microbes on you, even if you're sitting in an ambient plot, you have slower decomposition rates than you do if you have ambient microbial communities on there. So that was exciting to us because we think that really shows that these communities are not functionally redundant. And not only are they not functionally redundant, but they're having a signal that's similar to the signal we're seeing of being in a drought plot. Okay, so for those of you that are interested in particular microorganisms, I'm just going to quickly go through two brief slides about the diversity that we see in these plots and in these litter bags, because I think there's a couple fascinating things. So this is just a bar graph of different families of bacteria in the ambient plots, the drought plots, and the nitrogen plots. And one thing I want you to look at or see is that there isn't much of a big shift. So we're not talking about giant turnover in communities. What we're talking about is a shift in the relative abundance of just a few taxa are having this effect of composition. And in particular, there are 3 taxa, OTUs, defined by sequence similarity of our sequences, 3 OTUs within the actinobacteria that are really making up most of the abundance, not most, but a large abundance of the bacteria that are there, or microbacteria, and that's what's changing in these communities. And the same sort of pattern we see in the fungi. They're dominated by just a handful of taxa. I mean, you think a lot about microbial communities as being super, super diverse, and these are diverse. We can keep on counting sequences and we see new things all the time, but actually for the fungi especially, it's really clear, like the top 20 OTUs make up about 90% of all of the fungal abundance in the community. Okay, then here I think these data here then show us that there is this important indirect effect here of microbial composition on physiology and ultimately the decay rate. But this is only one piece of the story, because this tells us what's happening sort of initially as we change composition. But what happens over time? How important is this going to be as we keep running these experiments out? And so that's what we did. Next is we wanted to ask how fast does composition shift between the two environments? Because maybe it shifts right back, and at the same time we're running the experiment trying to understand whether composition matters, composition is changing in our litter bags, and it's probably being selected for by the plot environment that's there. And so the answer to this question will give us some sense as to the compositional resilience of the microbial communities. So the data I showed you were from the first year of our experiment, where we inoculated new litter bags in December. That's the beginning of our rainy season. We picked up those litter bags in March and June again, and then the experiment ended in November or December at the end of the dry season. So what we then did was we took an inoculum from these litter bags and inoculated new litter bags. And the reason for that was because as leaf litter is decomposing, the composition is changing too. So we sort of wanted to control for that succession. So every year we inoculated fresh litter bags, and this litter was collected all at the beginning of the experiment. So sort of the same litter pool we're using throughout the 3-year experiment. And we repeated that new inoculation again a third year. And so then I'm going to talk to you about the composition of the litter bags, which we took samples in March, sort of right in the end of the rainy season. You can see the dry season is very long here. So we have a short rainy season. We took compositional samples at the end of that. Okay, so just some background in terms of the diversity that we see there. There's 2 points I want to make with this graph. One is that we see that our inoculum is more diverse than what we end up detecting in the litter bags in March, and I think that's a good, that the diversity has decreased by the time we sample the litter bags and they've been out there after 3 months. And I think this makes sense, because you would expect that the inoculum, the initial inoculum from the environment, it is leaf litter specialists on there, but there's also soil bacteria that might not be able to decompose leaf litter. There's microbes coming from aerosols, there's plant microbes, they're all mixed in there, right? So we're giving our community a really selective environment. If you don't degrade leaf litter, you're probably not going to survive in there, right? So I expected a drop in diversity. The other point I want to make about this is that actually I find that very comforting, and especially comforting that we see that the 2011 samples are a little higher than the 12, than the 13, so the selection is ongoing, and to me it also suggests that our cages are working, that we're not having new microbes inoculating the bags constantly, and so we see that, and we see the same pattern for the bacteria as well. Okay, so to talk to you about the resilience over time, it's a lot of data that I have to boil down to ultimately one graph. So I'm going to first show it to you in terms of a MDS plot, because hopefully many of you are familiar with looking at these. So what I've plotted on the left here then is all of the samples, the compositional samples of fungi in 2011. And I'm coloring the samples depending on which, what is the microbial origin, which plot they were initially inoculated from. And so you can see there's a really strong effect of microbial origin after 3 months in the litter bags. You still see that the drought inoculated communities are different than the ambient communities. In contrast, if you have, this is the exact same points, you can see they're all in the same place, but now I've colored them by plot environment. And so you can see there might be an effect of plot environment, but it's much less strong than the microbial environment, right? Okay, so now let's jump ahead 2 years later, and we see sort of a different trend here, whereas it's harder to find a microbial origin effect, and it's a little easier to find a plot effect. Okay, so now to look at these different effects, I'm going to plot again the estimated variation for each of these, the 3 different factors over time. Okay, so that looks something like this. So this is the drought experiment, and just focus on the orange line. This is the effect of microbial origin on fungi over time. So initially there's a very strong effect that you saw in the distinct colors separating. In 2012 we actually don't see an effect, a significant effect, but there is one that persists in 2013. So there was some resilience of the community, and that it starts to look more like the plot environment that it's sitting in, and have less of a, well, actually I should say, there's some resilience in that the history of the inoculum is starting to count less and less, right? It's the composition is changing over time. But it still persists in 2013. If you went out there, you can see an effect of where that inoculum came from. At the same time, there is an effect of the plot environment, which we had hoped to see, and also the litter origin. And I was happy to see that these are pretty constant here, especially the litter, right? Remember, I'm taking the exact same litter every year and giving it to the microbes. And I hope that the same amount of variation is explained by that leaf litter every year. And it is, so that was good to see. So these are the fungi in the drought experiment. Here's the bacteria in the drought experiment. So that orange line, that's the most interesting line for me, not significant in all 3 years. So that means even though in December I inoculated with a particular bacteria or microbial community, by March, after 1 rainy season, I cannot tell where my inoculum came from. So that's highly resilient community, I would argue. In contrast, the plot environment had a really strong effect on the community composition of the bacteria, so they are being selected really quickly for which environment that litter bag is sitting in. Okay, so this is then talking about compositional resilience. What about the functioning in those bags? Because we saw that in the first year, immediately there was a functional effect. But how does that functional effect of microbial composition change over time, or what is the functional resilience of these communities? Okay, so you've seen this left-hand part of this graph, which is this is mass loss again over time, and so there's more and more mass loss over the season, and plot had an effect, and it still has an effect in the 2nd year and also in the 3rd year. So this is that is good. In general, leaf litter in the drought plots degrade slower than in control plots. Litter origin did not affect decomposition rate in any of the years. We don't see a significant difference across these years between the 2 trees where that litter came from. And microbe origin, while it did have that 1st year effect, as I've shown you before, by the 2nd year, it didn't have an effect. There was no difference between decomposition rates depending on where the microbial community came from. Right, so overall then it looks like that, that functional resilience is pretty quick in these communities, in that by the 2nd year, we don't see any difference in functioning based on the microbial inoculum. Okay, I'm going to just really briefly go over the nitrogen side of things, just because I think it's there's a couple points I want to contrast later on. The first, So we also see that the fungi in the nitrogen experiment, you could see a difference in the first year, and by the third year, that difference was still detectable. And interestingly, there was a slight difference here in the bacteria in that we did see an effect of bacteria that came from N addition plots versus ambient plots, even after the first 3 months. So in this case, we saw less of compositional resilience for the nitrogen effect. But in terms of functional resilience, we saw very little functional effect of these different communities. So even though nitrogen selects for different communities of bacteria and fungi, and they're very distinct, and they persist at least a year, maybe 3 in the case of fungi, we saw no differences ultimately for decomposition rates of those 2 communities. So in what way you're different then is important for whether composition matters. So actually, we do see a minor functional effect in that if I run this analysis with all of these nitrogen communities from the nitrogen treatment give a small percentage, and it happens to be significant because we have so many replicates. But that effect we can only see after we combine all 3 years. So I don't want to say that there's no effect, but it's pretty hard to imagine that by the 3rd year that anything is going on there. So the conclusion of this part of my talk, which don't worry is the major part of the talk, is first that we do see that these compositional shifts associated, at least with drought, are functionally relevant, but interestingly do not seem to be with nitrogen addition. Bacterial composition seems more resilient than fungal composition. And I thought was sort of expected. And one possibility, of course, is that the fungi just are not turning over as fast as the bacteria. Maybe they're persisting in terms of DNA and they're not actually even growing there, and we're just picking that up. In terms of that latter explanation, I don't actually think that's what's going on, because we do see huge changes in abundance over these seasons of fungi. So I actually think there's quite a bit of turnover. But bacterial composition, perhaps they're just growing even faster. And finally, community function appears to be more resilient than the composition itself. Or another way I kind of think about it is maybe it's the bacteria that are driving the decomposition in this community, because even though we see a persistent effect of fungi, the decomposition rates seem to be tracking more when the bacterial composition has shifted. But more to be done on that. Okay, but those conclusions then are great, and that's usually where I have ended my talk. And we've been working really hard to get beyond that, because the first question somebody always asked me was, okay, so composition matters, but what about composition matters, right? And that would be great to know. And so like other ecologists, microbial ecologists have been thinking a lot about traits lately. And so we've been trying to apply a trait-based framework for our ideas here. And so I just want to introduce you to a couple of concepts that some of you may be familiar with, but maybe others not. And that's in thinking about maybe 2 different types of traits, just for our context here in terms of environmental change. The first type of trait, broadly, you could think of as an effect trait. And this is a trait of an organism that affects some ecosystem process that you care about. So maybe it's nitrogen fixation, or maybe it's I'm going to talk to you about, I'll give you a clue, carbohydrate degradation. And one thing that microbial ecologists have that's a great advantage is we almost always have our phylogeny of our communities at the same time. And so it really helps us to think about these ideas in a phylogenetic context. So if you have some effect traits then might be really deeply conserved, like nitrogen fixation, for instance, or well, especially oxygenic photosynthesis, for instance. And so it might be that you have a lot of predictive power in terms of if I get a sequence of a bacterium and it groups with this deeply branching clade, then I have some idea of what function it's performing in the environment. So it would be great if effect traits were really deeply conserved. On the other hand, the trait might be totally randomly distributed across your phylogeny. And a lot of microbiologists have argued that this is probably the case for a lot of traits. And that would be because bacteria and archaea are so good at transferring horizontally genetic material, right? So you might not see a signal of traits on a phylogenetic tree. But that's kind of an open question still, I would argue. So first I'm going to talk to you then about effect traits that are related to our project. And we wanted to look at carbohydrate degradation. So you have your leaf litter. This is not our leaf litter, sorry. It's from a forest, obviously, not a grassland. And a lot of leaf litter is, the major component of it is carbohydrates. So we'd like to look at a trait that is looking at carbohydrate degradation. So we have all these polysaccharides that are being broken down by different enzymes that the microbes carry. Now, a nice thing about this is that we have a pretty good idea of what's going on at the molecular level, and glycoside hydrolases are a huge group of enzymes that are responsible for the breaking of these bonds of carbohydrates. And another great thing about this is that a lot is known about different classes of glycoside hydrolases and which substrates they're targeting that they might be targeting based on their genomic sequence, but also their predicted functioning. So we had, my husband Adam Martiny had an amazing postdoc, Renaud Berlamont, and he was a protein chemist that came to work with us on this project and did a lot of background work on just how we could classify different glycoside hydrolases, identify them, and classify them into these different substrate categories. Okay, so we're going to be classifying glycoside hydrolases based on the substrate that they have of the polysaccharide, or in one group, the oligosaccharides. And so, but each of these substrates then has a variety of families that can target them, and they're not homologous with one another. So the hard work was Renaud figuring, working and building upon the work that has been done by a lot of people before, and identifying these different families and grouping them by substrate. Okay, so I'm just going to I'm going to show you an example first in looking at the cellulases. So there's a variety of glycoside hydrolase families, and they all target the cellulose for degradation. Okay, so our first question was just remember that effect trait on a phylogeny, is it phylogenetically conserved? If it's not, then we're going to have a hard time putting this, trying to predict function here. And so Renaud found that they are indeed conserved, and what you see here, inside the circle is a phylogenetic tree of bacteria, and they just took fully sequenced genomes available, publicly available, and asked which of these genomes had the different glycoside hydrolase families. And I hope I'm not getting this backwards, but red means that that taxon had one copy of the family, and blue means it had more than one copy. So sometimes there's many copies of the family. And so if you just kind of squint your eyes, which you should be doing at this point in the talk, you'll see that there is some patterning here. There's some conservation, but of course it's variable depending on the family and so forth. Okay, and this is also the case, looks like it's also the case in our communities, in the sense that if you know about taxonomic composition of the bacteria in your sample, we think we know something about the glycoside hydrolases that are in the sample. So here are the different cellulase families, and Renaud analyzed some metagenomic samples from our leaf litter, and what you see is this is just categorized by the closest genera, and then those genera vary greatly in which glycoside hydrolase families they carry and how many of those genes within each family they carry. And you can go back and look at the number of observed cellulases in in a metagenome, and you can compare that to what you would have predicted based on the taxonomy, and it's a very close line. And these black points here are separate samples that we use to test this relationship independently, and they fit very well in this line. So all of this is to say that we think that taxonomic composition of our communities tells us something about glycoside hydrolase content in our communities. Okay, so that gives us some sense of the effect traits, and maybe it's, you know, it's not deeply conserved for sure, but it is somewhat conserved in that more closely related taxa tend to have more similar glycoside hydrolase content. So now we need to think about, though, how are these taxa responding to environmental changes? Because remember, that's the other part of this, is that these communities are changing, but they're changing in different ways depending on the environmental parameter that we're shifting. And so your response, whether you want to call it a trait or just generally a response, whether or not your abundance of an organism is going relatively up or relatively down, the next question is how that is related to the effect traits. So again, the response traits might be randomly distributed across your phylogeny, or they might be conserved. And not only might the ideal situation is they're conserved and they're correlated with the effect traits, and in this way then we could sort of multiply the effect by the response and you know, estimate a functional process rate there in my dream world. But we actually have no idea, at least when we started the project, whether or not responses would be to these types of treatments would be phylogenetically correlated. So this was the task of a postdoc of mine, Anthony Amond, who's now a faculty member at University of Hawaii. And let's just look at one panel here. And what he's showing here is that if on the x-axis is genetic distance between two pairs of fungi in our community, and on the y-axis is whether or not that pair is responding the same way to drought. And so the filled in symbols here mean that they're significantly positively correlated above this red line. So if you are less than about 8% similar in your sequence similarity, you're significantly more likely to respond the same way to drought than not, okay? So I would argue this is some evidence for phylogenetic conservation of drought in a response in fungi. And we see the same thing for nitrogen, that if you're within about 5% of sequence similarity of 28S ribosomal RNA gene, that you also tend to respond the same way to nitrogen addition. And this holds for both fungi and bacteria. And although you can't really compare these two genes because it's kind of like apples and oranges, it's still interesting that both of them are kind of around the same degree of genetic distance that we're able to see a correlation there. Another way to look at this, if you're used to looking at phylogenetic trees, would be again in this circle here. And on the left-hand side, I have nitrogen fertilization response plotted, and on the right-hand side, drought, and these are for fungi. And so what you should notice in this graph here is that if a clade responded by increasing in abundance, then that node is colored blue. And if it responded by decreasing in abundance, it's coded by red. And so you see that there's clustering of the red and blue around, right? So there are certain clades that respond positively and certain clades that respond negatively. And again, it seems to be around the same degree of phylogenetic conservation for nitrogen and drought responses. So that's cool. So then we thought, okay, can we do that multiplication of the effect by the response traits? Because now we have the response traits of our communities, and we can take the fully sequenced genome data that Renaud had and say, so we have some idea of some of these taxa, how many glycoside hydrolases we think they're carrying and which glycoside hydrolase families they're carrying. And so that's what I'm just showing here. So the problem in comparing these two very different data sets is that, of course, we don't have sequenced genomes for every taxon we see in our samples. However, I was surprised to find out we have 18% of our taxa cluster with a sequenced genome. So 18% of the taxa are at least, are no more than 3% similar sequence similarity for the ribosomal DNA. So knowing, because I want you to know it's a biased subsample, so these are taxa that we have a close relative already fully sequenced. Now we can ask with those taxa, if they increased or decreased in the treatment, what glycoside hydrolase genes did they carry? And this is a summary of a lot of work for Anthony of trying to do that. So using a correlation method that tries to take into account phylogenetic non-independence, he was able to then look at, okay, here's is there a correlation? There would be no correlation if you're on this dotted line here, the 0 coefficient value. Is there a correlation with the number of genes related to oligosaccharide degradation in a taxon and whether it went up or down in drought or nitrogen? And there's no correlation for this group. However, you'll see I'm showing the significant values in terms of the open circles. So if you see, there's actually 4 open circles here for the red points. So that means in 4 cases that we looked at, there was a relationship to whether or not you responded positively or negatively to nitrogen addition and what genes, how many glycoside hydrolase genes you were carrying. So for instance, taxa that responded positively to nitrogen addition also tend to have more cellulases. And in contrast, taxa that responded negatively to nitrogen addition tend to have fewer glycoside hydrolases related to xylem degradation. Okay, so it's a lot of material here, but the main point I want you to get from the 2 main points. One is we can find correlations with this method between effect and response traits. And 2, what totally surprised me here is we see most of these correlations for the nitrogen treatment, but not for the drought treatment, only in 1 case for the drought treatment. It was the drought treatment where we found that composition mattered, remember? So it's like, it was almost perfect, but not quite perfect. We can't bring the story all together. So why is it that we see in our experimental system that composition makes a difference for drought, but then we can't find it in terms of the genes we think that are involved in the actual degradation of the plant material? So there's probably a lot of reasons. I'm sure you guys could suggest a couple more I haven't thought of, but my favorite here, or the one that I think is most important first, is the biased subsample that we have of the genomes. Because remember, I told you, these communities are heavily dominated by a small group of taxa, and all those taxa are not represented in this analysis. So the ones that are most likely to have an effect on decomposition, I think, just based on their dominance, aren't all included in this analysis. So then, to conclude the second half and the end of my talk, that we see that decomposition potential, it's potential because we're looking at metagenomes, not actual expression, that's phylogenetically conserved and predictable by taxonomic composition in our system. That we see the response to our 2 different global change simulations is also phylogenetically conserved. And that there are some correlations between these 2, but it's not explaining our experimental results, so there's still a lot more work to be done. So I hope I can end here and convince you that there might be some potential for this trait-based framework where we combine a better understanding of the particular traits that are involved in ecosystem processes in terms of the responses, of those taxa to different environmental changes. And finally, and the powerful piece would be to bring those together with some experimental manipulations in the field and try to be able to tie all those things together. That, I think, would then really give us a way to start making predictions about when and where different types of environmental changes will really have an impact on microbial functioning in different environments. So thank you very much for your attention, and I have to thank all of my many collaborators, because this really was a huge collaborative project, and the students and undergrads and postdocs involved. Thank you very much. I'm happy to answer questions. Vincent, yeah.
So with your microbial cages, so you're excluding the role of immigration
Yes.
here. And so I'm wondering if you have any idea of, or tested anything already, like on how they're similar the response would be, like when you do this transplants, particularly like they see for a year, basically there's a really strong influence on where the sample of origin was.
Of dispersal, or what is the influence of dispersal on our results?
So the fact that, in a way, maybe the surprising result that you have that, what the microbial community of origin, was it has such a long-lasting effect on the whole year.
Yeah.
Is to some extent surprising. It of course disappears, but I wonder, like, if with immigration, you think that would be a very different result?
Yeah. So we have one experiment just went out last week to look directly at immigration, where we're actually putting out sterile litter and asking how fast do different microbes land on the litter. And we have two different treatments. One is just sterile litter, and one is just sterile nylon membrane, because what happened in our initial preliminary experiment, which we didn't really think about, is, of course, as soon as it rained, then there was a lot of growth. So then the dispersal experiment was over because we can't distinguish between what's dispersal and what is growth. So we have both things out now in litter bags with membranes that have a slightly, that are slightly larger. So they're 20 microns. So they allow for dispersal in of the bacteria for sure. And so we're basically just looking at how fast they're coming in. The other way that a graduate student in Steve Allison's lab is looking at that is he's doing some experiments where he's mixing communities from multiple places and asking, that's not doing a good job explaining his very nice design, but he's mixing the communities and essentially in that way asking, if you put them in a closed bag, does it matter kind of how much variation you've sampled at the beginning to decomposition later on? And there he's sampling across whole different environments to see whether or not that makes a difference.